The final process of transcription, which is copying a segment of DNA into RNA, is elongation and termination. Elongation is simply when the chain gets longer, so let's talk about the process. Initiation has already had RNA polymerase come and unwind a portion of the DNA, which then gives us the template strand and the coding strand. The template strand reads in the 3' to 5' direction and is what is going to be important in the process of elongation. RNA polymerase moves down the DNA template strand, adding one base at a time and adding in nucleotides. RNA polymerase binds RNA molecules out of the complementary nucleotides. The mRNA product is complementary to the template strand. The difference now between the RNA strand and the DNA strand are that RNA has a U for uracil attached, whereas DNA strand contains a T for thymine. As the process is happening, DNA is being unwound in front of the RNA polymerase, and is also being rewound once the proper nucleotides have been added. As the RNA polymerase moves downstream, this is elongating the sequence. For prokaryotes, the process of transcription would stop here. Prokaryotes only have 2 steps to completing transcription, whereas eukaryotes will continue the process of transcription. In prokaryotes, by the time termination would finish, transcription would have already partially synthesized multiple copies of the encoded protein. Eukaryotes have another step to complete the RNA strand, which is termination. Termination is when RNA polymerase continues to transcribe until it receives a signal to stop. RNA polymerase will reach a stop sequence, or a termination sequence, which will tell them to stop transcribing. This then completes the making of the mRNA strand and it can detach from DNA.